So good afternoon. Thanks for being here in person as well as over Zoom. My name is Lily and I coordinate the Matrix N cluster, and we help organize these mental health and addiction rounds that are starting up again for the 2024-25 year. And we are meeting every fourth Monday of the month at 3:30 p.m. to 5 p.m. here in this room. And this year we'll have another exciting list of trainees and faculty that will present interesting translational projects. And this year, Dr. Trisha Chakrabarty, who is with us over Zoom, is helping me organize these rounds. And if you're interested or have a colleague that is interested in presenting, please feel free to email me. And this month, we're fortunate to have Dr. Todd Woodward with us here today. And he will present, he is a professor at the Department of Psychiatry at UBC and also an investigator at BC Children's Hospital Research Institute. And today he'll be presenting about cognitive, perceptual and motor modes of fMRI brain activity. And the proof is in the bold signal. So please join me in welcoming Dr. Woodward. And if there's any sound issues on Zoom, please let me know via the chat.
I need the screen sharing enabled. Okay, so I'm going to talk about fMRI. So put up your hand if you're already an expert or at least know a lot about fMRI or a little bit about fMRI. Nothing. Someone in my lab put up their hand for nothing. Okay. So, hopefully that's over. Okay, so as you may know, fMRI measures fluctuations in blood oxygen level dependent signals, so that when neural activity is required, it takes place, the oxygen is required in those areas, and the MRI can pick up magnetic differences between the oxygenated and deoxygenated blood. So it's not actually measuring neural activity directly, but it's measuring the blood flow or the oxygenated, deoxygenated blood flow around the brain. So luckily, these bold signal configurations are very, very reliable. And that's what makes studying fMRI really rewarding, because often in, I don't know about a social science anyway, we often have problem with replicability and reliability, but in fMRI, it's really rewarding that way. So what the goal is of cognitive neuroscience, one of the goals of cognitive neuroscience is to link these patterns with the respective cognitive perceptual motor processes, because as hopefully you'll see in a second, just knowing what brain activity is observable doesn't necessarily tell you what that brain activity is responsible for. And so how do we make this link? That's what I'm basically going to talk about this whole time. I'm really talking about methodology and thinking in the proper way. I'll also show a lot of evidence for linking particular brain patterns with what I'm going to call cognitive perceptual motor modes. So I'm going to start with what is normally the task of cognitive neuroscience, which is just to look at those brain images and try to determine what the function is of that particular network. These are the three easiest ones that I could choose. I don't want to make everyone try to answer, or I'm not sure what I want you to say, but just think about what type of process you're going through in your mind about what the function of those networks is. If anyone wants to say something, go ahead. If not, it's kind of a trick because the middle one's the default mode network, and no one really knows exactly what the function of that network is because it almost always deactivates for most tasks. Not all, but most. So those ones should have been easy. These ones might be in the middle type of difficulty level. Do you think you could say what the top network and the bottom network are quite different in terms of what actual cognitive processes they're linked with? But just think about what you're doing right now. Like, you know, for the first one, maybe you felt quite confident. This one, medium level of confidence. These three are, I think, quite difficult. And I can tell you that they're very different types of cognitive operations that are associated with each one of those. I like to draw the little diagrams on there because that's what we actually use to identify particular patterns, shapes like that. So no one wants to say anything. That's totally fine.
Just
Just
the
think.
first one back. There's only two choices there.
Okay, what do you say about that?
That's not.
Well, the second one's the default mode network.
Okay, that's right.
The top one is, you know, hopefully people recognize that as a sensory motor type of network. This would be a right-handed type of response. So there's left-sided frontal parietal type of activity there. There's a right-sided cerebellar activation. That's fairly obvious. But, you know, still, just because we see that configuration doesn't mean we always know what that network will be involved in, what it will be doing in different conditions. The bottom one, superior temporal gyrus, is sort of auditory perception. So that's the easy one. This bottom one's, it's actually quite difficult, but, you know, feel free. This one, reward. Yeah, see, now that's what you've just done there. We could call that a type of reverse inference because you studied reward. So when you see activity, you say that must be reward. But of course, what goes on in the actual neurons is not necessarily what's going to happen with the blood flow. And so how do we, you know, I guess we'll talk about it a bit more. But anyway, this one here is language. So this is basically Werner's area and Broca's area. This one I won't talk about too much until later. And these ones are all different types of executive function. But anyway, you don't believe me? I'll have to show you the evidence. What's the proof? The proof is not in the image. The proof is in the bold signal. And so how can we prove these links between the activation patterns and cognitive perceptual motor processes? We can do like you just did in your minds and speculate by looking at the actual patterns. Sometimes the speculation is fairly accurate, but other times it's just complete guess. Or we can propose a function derived from task design connected to patterns, but that can be committing a reverse inference fallacy. Like when I started work in this area, my first paper was on anterior cingulate. We were studying the Stroop effect. So we said that the anterior cingulate was involved in conflict detection. Same time, other people were saying it's involved in error detection. Some people are saying it's involved in pain. Some people problem solving, whatever task the person used, they just said they were studying that cognitive process and linking it with the anterior cingulate. But what I put here is 6 different networks, and you can see it by these medial slices that something that people have called the anterior cingulate, but it gets into supplementary motor area, is involved in all 6 of those networks. So it doesn't, you can't really just link one area, not with fMRI anyway, not with the way the blood oxygen level dependent signal flows with one particular cognitive process. So what we really need, according to Poldrack, is to not have this reverse inference problem, is basically a one-to-one mapping between a pattern, an anatomical pattern, and a cognitive process. So the good news is that the anatomical patterns are very easy to identify. The cognitive process is much more difficult. And so I'll show you evidence about how we use the bold signal to determine what that cognitive process might be. Hey, busy slide. I'll talk about it anyway. Anyone that wants to interrupt me and have a discussion, just go right ahead. How can we prove these links between these activation patterns of cognitive perceptual motor processes? Well, first of all, we have to identify the patterns that replicate over many tasks. So that I will show you evidence for that. And it's all based on those little drawings, stickman and circles that I showed you before. As long as a certain combination of not just the anterior cingulate, but particular patterns in various areas emerge, we're very confident that we know what type of cognitive process is being, in a general way, is being carried out with that pattern. Then we propose a function or mode derived from the study how the bold signal changes in response to a wide range of conditions. So just imagine that if we can say the same pattern over many, many tasks, all of those tasks often have experimental conditions built into it. So the tradition of cognitive neuroscience it comes from cognitive psychology, where there's design of different tasks to study different cognitive operations. But now we find that a range of cognitive tasks will elicit the same patterns. So now we can study how the bold signal changes over a range of these different tasks to determine what the function might be of that network. And we can make comparisons between the tasks and within the mode. So if we have a mode that's, say, language, but we have that particular set of patterns that emerge for what we call the language mode in 6 different tasks, we can, within that one mode, compare across various tasks how that activity differs. Or we can compare between modes within one task. So if we had a semantic association type of task, that task might elicit 5 different modes. So then we can see, oh, this mode is not even active at all until the end of the trial, probably to do with response. This mode is active right at the beginning, maybe some type of early process, and there's different timing information for each mode, and that can help us interpret as well. And so if we can do this over time, so we can see these same patterns emerging in many, many tasks, then hopefully we can start to collect a specific set of observed associated cognitive, perceptual, and motor processes. So we can, this mode type of process will transcend just an individual task, and that we should be able to identify eventually a mode that can be applied to any task or to a variety of different tasks. And so this still requires words, unfortunately, so it's actually very difficult to avoid saying things like attention, language, response. We want to be kind of agnostic and think and not say this is working memory, this is reward, this is attention, this is avoiding conflict. We want to think of we want the data to tell us by looking at all these different tasks and when the modes are active or not. We want to have a set of words that actually doesn't have to be a study of working memory. It doesn't have to be one of the psychological terms that we've been taught to focus on. Okay, so this is Chantel, Percival, Linda Chen, and Hafsa Zahid. So over the past few years, we've been working a lot on this whole endeavor of identifying these different patterns that emerge over many different tasks and coming up with words that try to fit type of mode that applies to all these different tasks. So there's actually, now have identified 11. There's 12 here because there's one-handed response and two-handed response, but really that's just the response network. So some of them are probably well known already, like the default mode. There's actually 2 different default modes. We call them A and B in the literature. And some of them are actually quite new. So I'll try to give some evidence for what those are. So where does the evidence come from? So now let's talk about what fMRI gives us, which is amazingly under-discussed. To me, it just amazes me that I can go to a human brain mapping conference and for 5 days talk about the brain images, but never do we talk about the actual bold signal, the response that we're actually studying. This is what we're studying with fMRI. So how do we study these bold signal changes, and how does this provide proof or evidence for the cognitive, perceptual, motor mode that is induced for a particular pattern? So let's say that we wanted to know the bold signal in one voxel, so in just one area in the brain where there's no one knows how many neurons, but just the smallest possible area we can measure. If somebody screams at you, that one voxel may form a perfect change of the bold signal. This is sort of the hemodynamic response that's used in models of how the bold signals affect in various situations. So this would increase around 7 seconds, come back to baseline around 13 seconds, and there's just an undershoot there. So that's the pattern. And I literally, yeah, Tony.
What is the volume that's reflecting? Say a pixel, how big is a pixel?
Well, actually, this is taken from MATLAB code from SPM, statistical parametric mapping. So this was developed on animal models of the hemodynamic response to just a quick stimulation of visual cortex. So volume, I don't know, but in SPM and in many places, you can get the mathematical formula that produces the shape that came from that line of research. So I didn't do any research to get this. I took about that much MATLAB code from SPM. And I told it, what if you present something at that time point, what does the hemodynamic response shape look like? And that's exactly what it looks like.
So we don't know whether that reflects 1 square millimeter or 1 square centimeter.
Well,
in terms of specifying the area.
I mean, what you're asking me about is an area of research that I didn't do.
Okay.
Right? Like it's you would in theory say that is sort of like the smallest area that would actually have bold signal flow to.
I guess what I'm getting at in a way is what is the sensitivity of this technique in order to infer activity in a particular part of the brain.
Right.
Let's just say that
okay.
If it's 10 inches, then it wouldn't be very informative.
Right.
But if it's 10 millimeters, it's highly informative.
Yeah. Okay, so that's separate than this,
Okay.
but let's talk about that.
Okay.
That's super important. So what I've come to over sort of the last 20 years of doing research in this area is having to accept that the link between the way the bold signal flows and the patterns that create it is much easier to link to behavior than it is to link to neurons.
Then we're in trouble
Well,
because we're trying to make an inference about how the brain works.
well, I'm not necessarily.
Okay.
I'm making an inference about what type of thought processes, cognitive, motor, perceptual, are associated with the particular patterns of bold signal flow, right? The very first thing I said in the talk was that fMRI does not measure neural activity directly.
Well, that's another one of its problems.
Well, that depends on your perspective. From your perspective, that's probably a very big problem because you are studying neurons, right? So you're like, I'm studying neurons. fMRI people, are you studying neurons? We're like, we're not really studying neurons, but you're not studying neurons. What size? What are you looking at here? I'm saying I want to give up on that just for today at least.
Okay.
Yeah.
I won't interrupt any further, but it's a point for further discussion. at the end.
I am totally happy to talk about it. And I'm just willing to, put that forward that there is a perspective where we look at the association between patterns of bold signal activity and what is when that particular configuration of bold signal emerges, what are people thinking? Can we say? I think we can with very great confidence.
Yeah.
Yeah. You're confident in that too?
No, but the whole purpose of this is called non-invasive brain imaging. Okay? And if you want to interpret it the way you have it, it's fantastic. Your work is wonderful, but I'm just trying to get to the link between what we're seeing and what's happening in the brain and how we make conclusions about
The brain,
and cognition.
about neurons.
Of course, that's what's
Yeah.
in the brain.
Yeah. Well, bold signal is also flowing in the brain. So,
Well, I have a further problem about bold signal because, as you said, all it is blood flow. How do we make any inference about the underlying neural activity? And you've solved that by just eliminating.
you're preaching to the choir. I'm not even trying to do it.
Good. I'll do
So Tony is putting forward a view that probably a lot of you share, which is that if fMRI doesn't measure neural activity and you can't tell me how bold signal links to neural activity, what the hell are you doing standing up there with a group of people that want to talk about the brain, right?
and using a multi-million-dollar instrument to do it.
You're a little bit annoyed, right?
I'm not annoyed. I'm just puzzled.
Puzzled, but it's like, I think we should actually talk about it. So who's with Tony's? Like, what the hell is fMRI about? Nobody.
Good, I'm
Why
sorry.
not? Why not? I'm with Tony. I'm with Tony. I have sort of come to the conclusion that, like Tony's saying, it's just that Tony is basically thinking that I wanted to study the brain, but I don't.
Your field wants to study the brain.
Sure, I know. And I've never been very good at fitting in with my field, but I want to do what cognitive neuroscience, the goal of cognitive neuroscience is to associate a pattern of brain activity, but not neuron bold signal. It is a configuration of bold signal. When it's a particular way, what are people thinking? What type of cognitive process? That's all I want to answer. Oh, sure, Walter, sure.
If you ignore the image, if you ignore the image and think only about the time series, then it makes sense. And as Todd is saying, it's just a bold signal. It's a signal. I'm measuring my heart pumping here from my wrist, right? And it's some sort of signal. It's not my cells stretching in and out. It's something else that is somehow super indirectly related, but still works.
Hey, I can look at the images. I want you to look at the images. I still want everyone to look at the images. It's just the images are the way the bold signal is configuring into patterns. It's a beautiful thing, very, very reliable.
Usually time series.
Time series is something that no one ever talks about.
Yeah.
Because for some reason, even though that's
Put the
exactly what
images
we're meant.
back up. And what about the one in visual there in the middle of multiple demand?
Yeah.
What do you think isn't that leading us towards a conclusion that the visual cortex is involved in that process?
Well, yes.
Why would you put it onto an image of the brain if you didn't want to draw that conclusion?
Well, I can't help it where the blood flows.
Anyway, I don't want to.
It's like it flows there, Tony, so I guess that must be involved in visual activity. But it's just that if you ask me what volume is it,
Field in assumes that what you have is an indirect measure of activity in the visual cortex.
But not everybody. We can't speak in absolutes because I don't.
Very true. Anyway, enough of this.
I thought that was good. Okay, so here we are. Here's how the mathematical formula for bold signal change in response to one event, that's what it gives us. That's not actually a measure of brain activity. That's just a mathematical formula. And literally, I plotted that by taking that code from MATLAB and creating some numbers in Excel, which I just plotted.
What is the y-axis? What do the numbers mean on that axis?
The numbers mean nothing. It's just basically numbers that come out of a model, and I just change them. It doesn't really matter.
Well, don't they conventionally mean the change from a baseline in terms of percent of blood flow change? Isn't that what it is?
It could mean that. In this particular case, it's just in the mathematical formula. Yeah. But we'll talk a lot more about it. So if you have 4 people yell at you, or one person yell at you 4 times, each one of those times in one particular, the mathematical equation for bold signal increasing and decreasing, there would be one for this voice, one for this one for this one for this one, but they overlap. But of course, each one just produces its own actual change in bold signal. Okay, but what we actually get with fMRI is not 4 nice individual changes in bold signal, but we get this black line. So, this increases, starts coming back to baseline. Then when the second voice is heard, then it just kind of increases again, starts to try to go back to baseline, but because of the third voice, it doesn't get anywhere close to baseline, goes back up. So what we actually have is that. So what we get off the scanner is actually, like, for one voxel could look exactly like that. This is literally taken from MATLAB, from SPM. So this is with no noise whatsoever relative to what the actual mathematical model is for 4 voices coming at those time points. So how do we get from that to the underlying? Well, in fact, this would be more like a real type of experiment, where in this case, there's 31 different voices. Someone yelled at you 31 times. And so the bold signal is very, very complex looking. So it looks like a complete nightmare. But in fact, it's dead easy to get the bold signal actual increase and decrease shape that produced that complex set of changes. But for some reason, it's almost never done. And the way you do it is this finite impulse response model. So I probably don't need to explain that unless somebody really wants me to. But anyway, it's very, very easy. So what I literally did with this is I put into that mathematical formula in SPM, that MATLAB code. And I said, I presented a stimulus at this time point, this time point, this time point, this time point. So I just put it all in there, like a big timing vector type thing. And then it just gave me that exact shape of bold signal changes. And then I put this as zeros and ones, which I'm not going to explain exactly how they're set up, but I put that into, since I did this in SPSS, I didn't do an SPM, not statistical parametry, SPSS, like the psychology, tool that you use to do a regression. There's 10 independent variables of these zeros and ones. There's one dependent variable. It's all the numbers that came out of that MATLAB code for presenting here. That shape goes dependent variable, these 10 independent variables. The beta weights from that regression, it's a univariate regression, one dependent variable, the bold signals change. Those 10 zeros and ones, sets of zeros and ones as independent variables, 10 beta weights come out, you plot them, you get that shape. Perfect. It's easy, so easy. But for some reason, it's almost never done. Literally 98% of the time, nobody ever studies the shape that underlies the bold signal that comes out of the scanner. Moreover, let's say that you have someone yells at you for a short amount of time, longer, short, longer. Now you have 2 kind of long bold signal changes from that one and that one, and 2 short ones from the shorter one. Okay, so you see the blue one's kind of short, the green one's kind of short, the orange one's longer, purple one's longer, but what you actually get coming out of the scanner is that shape, that black shape. All you do, how do you get, what types of events went into that, or what types of bold changes underlie that complex signal? Again, I did this with the MATLAB code from SPM, which is literally 1 line or something. And I put in, okay, we had a short yelling at 3 seconds, 16 seconds, 23 seconds, but we had long yelling at 9 seconds, 12 seconds, 28. So that MATLAB code gave me that pattern. And then I take these zeros and ones, but now I have 20 independent variables. Before I had 10, because there was only one thing that caused one shape. But now I want to allow two shapes to emerge. So I have one set of zeros and ones for short and one set of zeros and ones for long, because in advance I take the yelling voices and I bin them into short ones and long ones. So with those 20 independent variables, now you have a beta, 20 betas instead of 10. Before we saw one shape, there's 10 points, 10 betas that produce that shape on the last one. Now we have 20, so now I have two shapes. Look at that, perfect. And this is, you know, with no error, but just using those zeros and ones, I was able to find the two shapes, short and long, that actually produced, according to the MATLAB code, that complex shape. So it's very, very easy to do that. So with a couple of little tricks, what we end up doing is finding patterns of bold signal change that are associated with particular anatomical patterns. So every time we do any task, like in this case, I don't know, a lot of you probably know Nicole Sanford. She finished her PhD with me a few years ago and now works here in the psychiatry department. But in this case, it was a very simple task where you just remember either 4 or 6 letters for 4 seconds or 0 seconds. And then you just respond whether or not you saw that in the previous set of letters. So in this case, we get 4 different modes that emerge instead of not like one brain image, but 4 distinct dimensions of bold signal activity. And each one of those is associated with its own bold signal changes. And so in this case, instead of 20 betas, these aren't actually betas because it's kind of a long story, which I probably won't have time to go into. I'd love to, but we ended up, you know, there wasn't just 20 independent variables. Now there was 40 because we have 4 different conditions: long and short duration of delay, and also 4 and 6 letters. So low load, high load, short delay, long delay. We have 40 independent variables per person, and we can get all the shapes out of there that are underlying the very complex patterns that come off the scanner. So I probably won't talk in detail about all that unless someone wants to ask a question about it. I mean, I will, but I mean, I could talk for 10 minutes on just this one slide explaining what all these different modes are and how we can determine what the associated cognitive functions are associated with these patterns based on looking at the bold signal changes. So here is one of the anatomical patterns. Momeni is an expert in this particular pattern. She's the first author on chapter that we're putting together, which identifies we've done this now. So during COVID, we couldn't really use our big computers, so I was just sort of looking for months and months at these images, and we came up with, you know how it was, like sitting there in your home office looking at these images for years. And so we just made these little drawings, which I was ashamed about at first, but now my colleagues have encouraged me to just say it. I'm like, fine. So we have the crab claw here. See that? Peanut. Left lateralized triangle, right lateralized triangle. So we give them names. Apologize, Tom. So here, this is I think based on probably average of two different tasks that we sort of ran early 2000s. But since then, we've finding the exact same patterns in various, like these are 2 completely independent studies that find the exact same patterns. So the top one comes from Toronto. This other one is collected in Vancouver, but it's not overlapping with these ones. So you can see how similar the patterns are. So believe it or not, it's actually very easy to find these patterns. Okay, so now we go through the difficult process, and this, you know, that fun part's over now. Looking at the brain images, now comes the part of actually doing the work, and the work has to do with looking at what types of bold signal changes are associated with that pattern. So for that pattern with the peanuts and the lower and left-handed triangle, the right-handed crab claw, this is what we see in a Sternberg task when there's 4 or 6 letters or 0 delay, 4-second delay. So interesting hypotheses can be made about what we should expect to see about the bold signal from this particular task. Notice we're not, even though we can hypothesize exactly which patterns, not only generally like dorsolateral prefrontal cortex, but we're going to see a peanut, we're going to see upper lower triangle, we're going to see a crab claw. A, anatomically, we can make very, very precise hypotheses. B, we can say exactly what we expect to see. So I don't know if you were at the MRI suites research day, but we actually, because we have such specific hypotheses about the anatomy and the hemodynamic response changes, we were able to develop a method to see whether or not the scanner upgrades were actually making things better, making things worse. just by having this precise, good knowledge about exactly what we should see. So anyway, the difference between blue and red is the difference between 4 and 6 letters. And so we have 4 letters here, 6 letters here, but that's when there's no delay period. So people see the 4 or 6 letters and we immediately show them 1 and there's just yes or no where they saw that. But if we make a 4-second delay so that they have to try to remember like what those 4 letters or 6 letters so they can say yes or no when 1 comes up, you have much bigger difference between 4 and 6 letters. So 4 or 6 here with 0 delay is small, but much smaller than when we have 4 or 6 letters, a longer delay. Okay, why? This is the working memory network. That's what we would have said if we were making a reverse inference error. And then we did say that when we first published papers on this. But now we've seen the same network in many, many different tasks. And so it's not working memory, it's something else. So we're calling maintaining internal attention, probably not great words, but in order to come up with the proper identification of that mode, we need to look at a wide range of tasks. And then we can compare. So this would be a comparison within tasks, but between modes. So this is default mode network. It's actually deactivations, not activations. But for example, in this case, the difference between 4 and 6 letters is exactly the same as the difference between 4 and 6 letters at 0 delay is the same as between 4 and 6 letters at 4-second delay. So we can do these types of comparisons where we say there's something different about the maintaining internal attention network compared to the default mode network. Obviously, there's huge difference anatomically, but that's not going to tell us very much about what the function is. What we have to do is actually say, look, one of this is much more sensitive to the combination of load and delay than the other one is. So why? And then here's another network or another mode, which we call initiation. So notice that this one's peaking around 9-10 seconds, whereas this one's peaking around 13-15 seconds. So that's another clue. One's early in the trial, one's middle of the trial. There's also response one that comes late in the trial. Oh, it must be encoding, maintaining the information and response, right? That's reverse inference because no, it's not just those things. There's a wide range of different situations where you see the same network. So in this one, we do see greater activation. So we have 4 low load and high load. Low load is red, high load is blue, and here's red, here's blue. There's a load dependent type of effect. But an interesting thing that happens here is that when there's 0 delay, there's actually higher activity. I mean, it's not maybe obvious at first why that is, but it's just because this must have something to do with initiating cognitive processes, or maybe it has to do with the initial contact with the visual information. But we see 4 letters or 6, and then if there's no delay, that other letter gets put on immediately. So that's why when there's no delay, there's actually a longer response, just because when you take the letter away, when that's the letter to say yes or no to doesn't come immediately, then the whole thing just goes back down to baseline, doesn't come back up. And so we can compare to internal attention and this initiation. This peaks very early, has probably smaller load effect, and the 0 delay condition is actually higher activity than the 4 delay condition, and it's the opposite here. So this is the process. And this is what we spend all of our time doing in the lab, because now we've kind of finished with the anatomical side, and we have software that or some MATLAB code that actually classifies the networks, but you can eyeball it. It's just easier to go through a lot of them to classify, but we do check by making sure those patterns are there. So Lyndon and John Shackey and I put forward a paper, and that's just the hypotheses about what we'd expect for this particular task. So a whole paper on hypothesize with all of those little patterns for every single mode, and exactly what we'd expect the bold signal changes to look like for every single mode for a type of task that had 4 or 6 letters and 0 and 4-second delay. So you can see there's one that peaks early, one in the middle, one late. This is default mode network, kind of deactivates throughout the whole trial, and a different network that has to do that we call focus on visual features will actually get deactivated. So sometimes, even though you see activity in the visual cortex and you show the blood flow actually decreases to that area. How's that? And here's the anatomical for each one of these pictures here, or each one of these colors. Here's the anatomical configuration associated with each. Okay, so this is how traditionally task-based fMRI is done, and hopefully I can show you how easy it is to make a mistake. So normally what happens is that mathematical equation that I showed you that comes with that produces the exact mathematically perfect hemodynamic response change to a particular type of timing. That's not usually used like I used it as a dependent variable to see whether you can retrieve the pattern that was underlying that complex pattern. That's usually used as a model. You know, the idealized hemodynamic response change based on a particular set of task timings, okay? So normally what we see in a brain image is actually pretty much just the correlation between that model and the actual bold signal, which has a whole bunch of noise and, you know, different types of sources to it. And so normally what we do, and this I also took from the MATLAB code that I talked about, we create a shape based on a particular set of timing and say, okay, let's just correlate the actual bold signal with that model. And then the correlation is basically put on the brain. So a very high correlation will get a light color in the brain image, okay? So the problem with that is that you really only get one model with a few different options for sort of fitting things that aren't exactly perfect to this model. So if we put in a model like this, which is very, very commonly done. So if we had a Sternberg task and we use this assumed model, that would correlate with only the blue areas of the brain. But we know that from many, many experiments over many years that there are actually 5 different modes that are active, all with different shapes of the bold signal change. But if we use this model, we're going to pick out that one only and miss everything else. So we miss, you know, 80% of what we had to learn about. And we conclude this is the working memory area of the brain, when in fact, there's a whole bunch of things that we completely miss. Or we might say, okay, we're just going to put in a model that goes the whole duration of the trial. So, you know, not just 4 seconds of coding, 2 seconds delay, just sort of 8 seconds worth of stuff, like a block design type thing. So then what you'd say is, let's see how much increase there was over the whole trial back to baseline. Well, what you might do, you might have sort of a partial correlation between that model and all of the actual bold changes that happen with all 5 networks, in which case you wouldn't be able to tell the difference between all the different modes. You would just have kind of one brain image with everything mushed together. That's also very, very easy to do. So you can, just by using that assumed model, we end up potentially or certainly making mistakes. We're not actually finding the configuration of bold signal that's associated with the different modes. We're just basically putting everything together. We're just picking out one little part, but definitely there's no way that you would get an exact match between what the bold signal actually does and the model. Why not just use a finite output response model, all those zeros and ones, and then the underlying pattern that underlies that complex bold signal split into dimensions will just emerge for you. You don't even need a model. This just tells you the bold signal changes that are happening around the brain for those particular images. So let's do that. So what are we going to do about this bold signal? It's very, there's numerous sources, very complex, but what we can do if we have a task. So, you know, fMRI is almost, well, usually resting state. So usually just people rest in the scanner, do nothing, and then you take their bold signal change over a long period of time. And that's a very complex type of fMRI signal that is emerged from resting state with, you know, worrying about being the scanner, breathing, heart rate, all kinds of issues have to be sorted out. But if you have a task, now we can actually extract the variance in the bold signal that's predictable from the task timing. And just by doing that, we get rid of a lot of problems, because the only type of bold signal changes that will be preserved are the ones that are related to the task timing. So unless movement or breathing or heart rate are actually related to the task timing, you don't have to worry about that with task fMRI. So that's what we can do, constrain the variance. Also, often combined with resting state, often people use regions of interest or you know, parcel the brain into, say, 180, 200, 300 different areas. So by doing that, we're forcing all the voxels within a parcel to be on the same network, which is, you know, just, it's impossible that you would actually choose a configuration that would be exactly mapping onto what types of correlated bold signal would actually emerge. So we need to allow, we had to get to the smallest possible areas that we have to measure with fMRI and allow them all to participate in the patterns. And this is how we get the specific shapes that I was talking about. We can hypothesize specific shapes, but we'll never see those shapes if we actually use partials, because the partials will just completely occlude things like a crab claw or, you know, peanut shape, things like that, because the beak might be too large. And finally, we don't want to assume this HRF shape. Like we've already talked about, this is, you know, to make an accurate shape is very, very difficult. We don't even need to put in a shape. You just put in those zeros and ones and it tells you the shape, and it's specific to a person, specific to a task condition, and specific to a mode. It seems to me to be a much better way to go. So maybe why isn't that used? I think it's just tradition. Normally, when people do task-based fMRI, they look at the correlation between bold signal and the model. But if the model is just zeros and ones, it will tell you what the bold signal is without making any assumptions. So we can get rid of these assumptions about boundaries and parcellations and about assumed shapes, and we can extract just the 10% of the variance that's predictable from task timing, and that's how we can end up with, you know, these anatomical consistencies, and also how we can look more at the bold signal changes. So I really like to go over all these different modes and talk about how the bold signal changes for each one of them. So that's what I want to talk about, but I'd be happy to talk about something else or answer any questions if anyone wants to have any discussion that comes to mind. Yeah.
Why are people so concerned about the power of the fMRI? Obviously, they are, because they want to get down to the smaller volume of brain. Why does that matter from your perspective?
The power of the fMRI?
Well, I mean, people want to have a 7 Tesla or
Oh,
13 Tesla or a
right.
3. And the reason for doing that is that you get a sharper image of both the underlying anatomy and a smaller area of brain that you're trying to infer is involved in whatever the signal means. What you're telling us, which is perfectly logical, why can't you just do everything with a 1.5 Tesla?
Well, actually, a lot of, you know, the difference between what we're looking at here and a 1.5 is not much. Like this is mostly done on a 3T scanner.
Yeah,
and
but
this,
I'm saying, why are people spending $10 million for 13T as opposed to $1.5 million for a 1.5?
Well, if you're studying the bold signal, it's not going to help that much, is it?
No, but that's my point.
And that's
People
my point
aren't
too.
just studying
Tony,
the
you're trying to
bold
criticize me, but I'm agreeing with you all the time. I agree with you, but it may be. So look, whether it's 1.5 or whether it's 3T or if it's 7T, this is the basic thing you're going to find. But if you have a 7T, maybe you can actually find more. You can find more patterns, or you can find more specific patterns. But I've been doing this for 20 years and started with 1.5, and I can tell you this is not even accepted yet. So until we start looking for the patterns in 7T, maybe we can just all agree on, you know, what is easy to find in a
Yeah, but I still haven't heard the logic of going from 1.5 to 7. If the identifying the anatomical locus is not even of interest.
But it's identifying the anatomical locus of interest is what's of interest to everyone except me.
Okay.
All right. So I'm not going to defend it, because that's not, if someone wants to move to a 7C scanner from a 3T scanner, I will say, I can tell you exactly what patterns you should see, what the basic patterns are, and exactly how the bold signals should change. So I would like to check from when you go over for the 3T, 17, 70, I would like to know if it helps me or not. I'm not saying I know it will. I'm not saying I want to do it.
Like you're making a fantastic case for this being a complete waste of time.
Well, if you want to study the brain, the neurons, it is a complete waste of time. And I think everyone already will agree that we're not studying neurons. Like, since when did people think we're studying neurons with fMRI?
Well, everybody in the brain imaging field thinks that they're looking at the brain, even though it's one step removed from neural activity.
Well, clearly we are looking at the brain, just not looking at neurons. Got to learn to embrace the bold signal. It's a beautiful thing.
It's very provocative, and I love the fact that you've been persistent. Your data is certainly internally consistent. Just I'm thinking of perspective of all of the field. And
well, field probably doesn't want to want me to say this too loudly, but I'm into it. So we're doing
I'm into neurochemistry, so I'm even 10 steps removed from the brain.
So you were discussing how the bold signal activity can't really correlate with brain, like neural activity. but I don't know if this actually makes sense, but I'm wondering if any comparisons have been made with temporal aspect with the EEG signals.
Well, temporally, it's really difficult to do that because look how slow the bolt signal is.
Oh, right, yeah.
Yeah.
That makes sense.
But anatomically, it has been, there's sort of evidence that they do overlap. But, you know, I would love to start this whole thing over with just EEG data, because there's probably different configurations of EEG signal, which is going to be a lot more complex just due to all the different frequencies that are involved, you know, very much smaller time scale. I mean, we have published some papers that kind of tried to determine the overlap between the modes or the anatomical patterns that we see with fMRI and EEG. But I think I think that, like we're sort of mentioning here, that even just fMRI and agreeing on what would be the thing to compare to is not set yet. So any other points for discussion?
Do the patterns change in different types of populations? Have you been able to, like patient populations, for example?
Yeah, I mean, obviously stroke, they change a lot. Schizophrenia, we see the same patterns, but the thing is sometimes the bolt signal changes are different. They might be shallower or higher, but that's just much more, it's not as reliable as seeing the patterns in the bolt. Like within a subject, we can change the bolt signal quite consistently and the patterns are very consistent. So with schizophrenia, we see the same patterns. And sometimes we see the bolt signal different change too, especially sometimes, like I have a slide here that talks about hallucinations and delusions, and particular networks seem to be different. For example, there's one of these modes is called the reevaluation mode. And so when it sort of peaks even after response. So we think that people are actually, the cognitive process has to do with checking back of what we were just doing. What was I just doing? Did I do that right? Did I do that wrong? Well, that's the one that's reduced activity and delusions. And as we know, delusions are fixed false beliefs. So if someone has a delusion, they don't usually want to apply that kind of reevaluative type of processing. And that does emerge. But you know, does that mean that we've identified particular neural damage in schizophrenia, or does it mean something we already knew, which is that they're just less willing to revise their beliefs? So if their thought processes are reduced, then maybe, you know, that's exactly what you'd expect to see in the bolt signal. So, you know, it's just an ongoing type of, I don't know, sometimes obviously if you have actual brain damage, if you have actual neural damage, then that will affect the way the blood flows, because when the neurons aren't firing, the blood won't go there. And so, you know, we did that Laura Boyd, and we published a paper on stroke where, you know, basically we couldn't really average over the sample because everyone had, like every person in the sample just had basically a flat bolt signal change because they actually couldn't fit the patterns because they had brain damage. But, you know, the sample of healthy people, then everyone just could be aligned together. So. Yeah, Walter.
It's in line with what she's saying, different populations and embracing the both signal, which is our case. Imagine if you can separate vascular dementia from other dementias. That's the case. But imagine as it's just perfusion.
It's fine. Yeah. That's the claim.
Yeah.
It's not. The claim is that there's some damage somewhere in the sub-region of the frontal cortex that gives rise to schizophrenia. So anyway, I like the entire field of neuropsychiatry.
Okay, so let's look at some bold changes. In this case, we would be comparing between modes for semantic association. Okay, so in this particular task, we have, well, you can't see that, but we're going to ask people to say which of these words on the bottom are most closely related to the word on the top. So the answer to this one is arrow. And the answer to this one is hen, but this was a more distant relation. So arrow, archer, I mean, they're very highly correlated, so that that's fairly easy. But fox, hen, I mean, yeah, they're both animals, but you don't really think of a fox and hen necessarily, you know, in kind of closely associated as archer and arrow. So we get various different cognitive modes from this type of task. This is what we call language mode. So we have patterns here that I would love to describe. So here's the disappearing face, two little eyeballs there, and a face and a smile. And so that is actually specific to this particular network. And then we call this rail shot. Like think of a pool where there's two cue balls. There's Broca's area, Wernicke's area, at least the fMRI version of them. And we draw a little line around there. So this is what we expect to see with language. And this one, we have eyebrows, and then we have this tear going down, but this tear is blown sideways, believe it or not. Like imagine if you use parcellations, you would never be able to see that type of specific pattern. So there we go. There's our language network patterns that's been published, at least pre-published. And then we have also during this particular task, this one that I talked to you about before, maintaining internal attention. We have some upper triangle, lower triangle. That crab claw is not that well formed, but it's very close. And overall, we have to, conclude that probably is the maintaining internal attention network. So here's Eric and John Shackey. They put this language chapter in Psych Archives, and this is the hemodynamic response, or the bold signal changes for semantic association task. So in this case, the distantly related words, so fox hen, have higher activity than the closer related words. So obviously, the more we need to work on determining what those semantic associations are, the more activity there is in that language network. But the and there's Ava, she's getting a chapter done here for the maintaining internal attention network. And we have the exact opposite pattern with that network, where actually the closely related words have higher activity than the distantly related words. So even though those two networks, those are two of the examples I showed together at the beginning, anatomically, you might not really be able to see the difference between them, but temporally, they're complete opposite in this particular task. Why is that? And I made Ava check it, like that we didn't have the labels reversed, because I just couldn't believe that was the case. But that's not a hypothesis that we had. That's something that we've discovered. So now we would hypothesize for that particular type of task for this network. If there's closely related words, there's more activity in maintaining internal attention network, but there's less activity in the language network under those circumstances. So, this is the unfortunate thing. We have to speculate now about why that is, because this is the first time we've seen it. So my speculation is that when we have, or our speculation, I hope you share it with me, is that when we have closely related words, you see archer arrow, you just start thinking, imagining an archer arrow, an archer with an arrow, and you start to focus on that internal representation, and you see more activity in this network that's involved in, Ava's paper that's been submitted to Journal of Cognitive Neuroscience has imagining the future, imagining the past. We see activity in this network in that situation too. Or when you're trying to think about a situation that's relevant to you, it all activates this network. So by going across all these different tasks, we can kind of try to come up with a particular cognitive mode associated with that pattern. So anyway, that's just the direct comparison of two different modes during the same task. And then, we'd be able to identify the patterns and then make conclusions about what that mode might be. So I have about half an hour left, or should I stop now and discussion?
Usually leave 20 minutes for questions.
Okay. I have lots, like I do longer, but so let's have even more examples. Let's see what I have here. Okay, so this is Nicole again. So for a paper we published in Journal of Cognitive Neuroscience. So in this case, it's not verbal working memory, but it's nonverbal working memory. So we have to remember where in the location those dots are, and then we see one dot, and there can be, I think, 1, 3, 5, or 7 dots, and then we see one afterwards after 1, 3, or 4.5 seconds of delay, and then we can see what types of modes are involved in this type of task. So here's an example of what we call auditory for response mode and the actual response network. So you can see similarities here, like there is some sort of areas related to responding on this particular network. On the left here, we call this auditory attention for response, but it has auditory and motor type of areas involved in this pattern. And this is the response network. We don't see the auditory, but we just see the response. So there is quite a bit of overlap. So you can see that the supplementary motor area here, some left lateralized type of sensory motor areas. But look at and this is within one task, within that non-verbal working memory task about the dots. So these two different networks, even though they have a lot of overlap, look how different their bold signal changes are. There's actually a suppression of the bold signal changes in this network, And that's delay dependent, 1.5, 3, 4.5 seconds. And then for the response network, of course, there's activation because people are actually responding. And of course, there's a staggered peaks here because the delay period gets longer and longer and longer. So the time when people respond gets later and later and later. So now we have to, this auditory temporal response network, why the heck is it reduced activity during this? So what that means is that there was more activity in this network before the trial than during the trial. There's somehow a reduction in the network. So, what was going on before, or does this network somehow have to be reciprocally activated relative to the response network? But we have to look at, many, many different tasks and many different trials to be able to understand why that happens in this particular situation. We don't have just an answer from here. We could just say something to do with the task, but that would be reverse inference error. Can't just sort of say whatever the task had to do with and say this is always involved in that, because we know that we'll see that network in many other situations. Okay, well, now this is an example of comparing between tasks for interpretation. So on the top, we have a lexical decision task. You can say, yes, that's a real word. No, that's not a real word. No, that's not a real word. And on the bottom here is what we call Raven's matrices. So we have to figure out what the pattern is. It's 1, 2, 1, and we would say yes for that. And then 1, 2, 3, 1, 2, 3, 1, 2, what's that? And the next trial, this triangle, so it should be a circle, so we say no to that. And this is what we call the focus on visual features mode. Okay, look at all that visual cortex in there. I mean, there's actually some deactivation in the actual primary occipital area there. And so there's a reciprocal activation within the visual cortex. I mean, then during the RSPM, so that's the task which involves Raven's matrices. We have to figure out the puzzle. What's the next one? There's actually strong activation. Why? Obviously, people are checking carefully what the pattern is and using that area of the brain to, and because those neurons are firing there, we're assuming then the blood flow is sort of collecting to that area and we see activity because they have to focus on visual features of that particular task. But why is there deactivation for lexical decision? Why could that be? They're still seeing things. I mean, I don't really know the answer, but from looking at many, many tasks, and we see this during a Stroop task, for example, let's say that when you have to ignore certain properties of the stimulus, we actually see deactivation of visual areas. So we're not really having to ignore information in the electrical decision task, but it's irrelevant what color the letters are or what they look like. It doesn't matter because you're just trying to say word or not word. So it may be that unless you're actually focusing on visual information, that you actually get deactivation in this network.
Well, that's really fantastic. So when you see the suppression, wouldn't a simple explanation be is that if there's two competing networks, and so for one of them to work optimally, the counteracting network has to be inhibited. Because it's interfering. If it were on stream at the same time as the one that's processing whatever the feature is, then you wouldn't get a very good correlation. But the fact that you separated them looks to me like reciprocal inhibition. It's removing something that could interfere with the proper performance of the task.
Well, but keep in mind that these are two completely different tasks on different people. So the network activates during the Ravens task, but deactivates during the other task, but it's not on the same people. So it's not just a simple type of reciprocation. It's actually a way to compare between tasks within a mode.
Well, I guess if you focus on the tasks, what are the features of the task, whereby whatever the feature of Ravens is, that network would interfere with the main process that's happening in the other network.
Okay, so yeah, okay, so let's just talk about one task. So forget about the Ravens, let's talk about lexical decisions because that's where the suppression is. So yeah, there's a language network in that task that emerges. So you're saying maybe the language network's reciprocally related to the
Visual.
Yeah.
So,
but other times they activate together. If it's important to look at the visual information to do the task.
anyway, just
That's why
a simple explanation that probably isn't true.
Well, that's why it's so informative to look across a whole bunch of different tasks, a whole bunch of different situations. So you see it's not always reciprocally related. Depends. And that's kind of the answer to what happens in special populations too, like in, correlative delusions. If you have, more activity in the language network for hallucinations, that does depend on the task, even task conditions. It's not just something where you say in schizophrenia, this network is overactive. It just depends. You have to replicate. It's much more difficult with special population just because we have to collect so much more data. But these, now there's data available online. You can just download, task-based fMRI data. We can compare across many different tasks for all types of hypotheses about these networks. Well, I've got more of that, but yeah. So I'm just sum up. So what we want to do is identify these anatomical patterns: the crab claw, the peanut, the what else I have there? We have the smiling face. We have the tears blowing sideways. See those replicating over many tasks. Propose a function derived from study of how the bold signal changes in response to a wide range of those conditions. Comparison between tasks and within modes, like we just did on the last one, or between modes within tasks, like we did in the working memory, where we can see that certain modes activate early, some in the middle, some later, to develop these connection. And then each replicating pattern collects its own specific set of observed and associated cognitive and perceptual motor processes. So if we can collect a lot of information, maybe we can come up with an interpretation of mode that's actually one-to-one mapping with the pattern. That's the whole point. That's what we want to work towards. And that's what's basically considered impossible in the field. Just because I think because of using the assumed models, people are basically getting different results all the time and don't see the common patterns that it would be there if we didn't assume a model, if we didn't take, partialations, if we just let the data tell us what there is to find. We find very, very reliable type of patterns. And that because they replicate over tasks, we can start to collect information about what the overall mode is that they're involved in. And so this one-to-one mapping may emerge eventually, and we're hoping that, and sort of feel like we're moving towards that, but obviously there's still a lot of data to be collected or downloaded and analyzed. But unfortunately, it's just very, very difficult to avoid words that imply things. In fact, like for example, we call one of the modes, let's see if we have one of the modes we call multiple demand, only due to pressure from reviewers in the field that this network, that the patterns match exactly the actual multiple demands network that's already known in the literature. That doesn't mean that I want to imply that when I see that network, that the type of processes that the multiple demands people said it does is what I think it does. I just have to use the word just because everyone say, well, it's a multiple demands network. Yeah, it's okay, call it whatever you want. And here's what it does under a variety of different situations. It is not always what they, came hypothesized originally, which is taking a difficult problem and breaking it up into sub components and solving each component separately, then putting it all together. That's reverse inference. That's because they were using the Ravens task. A lot of them use the Ravens task, early multiple demands. So of course they said that's what it does. But now we know that it activates, that same pattern activates many, many different situations that don't involve breaking down a problem into sub problems. Or at least if it does involve breaking down a pattern into sub problems, there are other networks that also emerge in the same task, other modes, right? So it can't be just that one mode that's involved in such a process. Anyway, yeah. So this is just the kind of what I was talking about, but it's just really the bottom line is that even though we published papers showing that certain networks are deactive in delusions, other ones are overactive in hallucinations, it just depends on what the task is and what the conditions are, and it's just very, very difficult to go from the cognitive mode interpretation to a full understanding of how those are affected in special populations. So I think I'll just end there and mention everyone in the lab that worked on this, and you can have a discussion.
Any other questions? I'll monitor the chat.
Luke?
When you see the sort of lines in your template shapes, like the
Shall I show one? Shall I show some lines?
Yeah, you had like a rail shot and
Rail shot.
Yeah, the wishbone say. is the assumption are the lines constrained by the cell count? Right, must be the. Do you think that's what's going on there? Or?
Well, the lines just emerged from my tired COVID brain, thinking that it looked like a pattern. But, you know, again, it doesn't have to do, I'm not trying to make any bold claims about biology. I'm just trying to say, we can see that if you see the smiling face and you see tears blowing and you see the rail shots, I can tell you what people are doing and I know what the hemodynamic response changes are going to look like.
Yeah, but aren't there, so that works there where you're looking, you know, where the lines are formed by the edge of the brain, but aren't there other brain areas where the so-called gyral morphology is quite different from person to person? So.
Oh, so yeah, we are averaging over people. Yeah. And, you know, SPM does that by morphing the brains into a common space. So, you know, how well we can actually find these patterns in individual people is a question that I haven't looked into close enough. We're sort of developing projects that will allow us to test. But so far, it's just been using SPM to normalize everyone to a common space and identifying these patterns that we can see averaged over everybody. And the hemodynamic response changes are averaged over trials. They actually, you do get a separate bold signal change for each person, but their anatomy is always constrained to the overall pattern. So the extent to which this is usable on an individual level is still.
When you say you can predict what tasks they're doing, you still mean on a group level, not on a.
Well, I mean that, yeah, I mean average over the group, but you know, it may still be, I think that, you know, we do want fMRI to be used on an individual, right? So the thing is, you know, how well can fMRI really do that? And so what we want to do is come up with a method that is not affected by noise. That's least affected by noise, because that's the problem with an individual person relative to 100 people noise, right? I think that this method is very robust in terms of not being sensitive to noise. So I think it is a really good way to investigate individual people, but it's only sort of just recently, we haven't even put all this out into the literature yet. You know, so, you know, we're working on doing that just to let people know these are the patterns average over people that we'd expect to see and what the mode should do under different task conditions. And then, you know, we have projects in the lab where we're working towards determining the reliability of that on individual, because that's where we all want to go with it, of course. And maybe that's why 7T is important, right? Maybe higher powered, there's more complex networks. I mean, this is sort of basic stuff. You know, it may be that you need machine learning to actually identify more complex patterns that are, you know, but, you know, at least we can find very, very reliable patterns, and putting funny names on it really helps people to identify them. And it's just, if you see the same thing in multiple studies, it gets more believable when you start to recognize the patterns. Anyway. Sorry, Tony, there's someone.
Somebody else.
Okay, Tony.
Thank you.
I was just wondering, so you've done this comparison across many different tasks, and you've noticed the same networks between the tasks. But for an individual task, let's say the Ravens, have you noticed that have you come to different conclusions with the patterns that you've been finding with your method of analysis, or has it been more that the conclusions being made through using one task to identify a phenomenon or something that they think they're identifying or looking for, you're thinking that's somewhat misformed based on?
Yeah, I mean, we have a whole series of papers that have been published using this method, and over time, slowly, we stopped making a serious reverse inference errors. But of course, I mean, we were lost 10 years ago. We had no idea about there was patterns that were replicating. So we would just call it whatever we were studying, whatever the task was, you know. But now I'm trying to avoid that. But I'm getting stuck still saying things like response or well, responses, maybe these one, but multiple demand or language, you know, like, or, you know, when Tony mentioned, look at the visual cortex on that one. Well, yeah, it's visual, but it's deactivating under many conditions. So it's not only visual, you know, it gets, so I don't know, that's kind of where we're moving towards, is really trying to avoid that. But it's very difficult, and it's just, you fall into a trap, because every time you say a word, someone just has all these ideas that come into their mind about what you're trying to say, which might not be accurate.
Are you getting significant pushback from like if you take data from somebody else's?
No,
data set?
not at all.
No.
Not at all. No, I don't know. It's just, but it's not pushback, but it's also not encouragement. It's just kind of, people are just like, don't know what I'm talking about. Just haven't heard of it before, I guess.
Is there something that, like, in your mind, ideally, you could demonstrate something that would convince the field that this is worthwhile exploring? Like, what do you envision that proof?
Yeah, I think that, you know, the type of thing that I've explained today, I think people just think it's like sort of it's just something that's not really important. I think that people won't pay attention unless we can show that, for example, we could predict, you know, relapse and schizophrenia more accurately than other ways that are available, or show close associations with behavior. I think that's kind of where we're going. We do try to develop that. So yeah, you know, nobody really wants to go through all this and go through all the tasks and look at all these things. It's like, well, you know, like Walter said, can it tell the difference between two types of dementia? That's what people want to know. So I can't do everything because basically we're the only ones doing this. So if just other people would try, maybe we would find that it is better, but it has to be tested.
Your questions?
Nobody online?
No questions. There
Okay.
weren't any people.
So
Thank you again.
thank you.